First of all, I would like to thank the organizers for providing me the opportunity to present you some part of the work we are conducting at Neuro. So I have no disclosure. The research unit I'm conducted is called Nutrition and Integrative Neurobiology. It's located in Bordeaux, here in France, and so we are mainly interested in this research unit in understanding how nutrition can influence brain function and vice versa, and whether nutrition can participate to mood and cognitive disorders. And today I'm going to talk mainly about long-chain polyunsaturated fatty acid I'm more interested in, and my group in this unit is working on this. As you probably know, polyunsaturated fatty acids are essential fatty acids, meaning that the body cannot produce them, and they have to be provided to the body by nutrition. And there are two main families of PUFA. The first one is omega-6 family, and the second one is the omega-3 family. So the precursors of this PUFA are found only in plants, and as you can see here, in different plants and oils, and like walnut, flaxseed, rapeseed for omega-3, and sunflower, corn, peanut for omega-6. Once we consume these precursors, then they are metabolized by the body through the same enzymatic pathway, meaning that the proportion of linoleic acid, the precursors of omega-6, and alpha-linolenic acid, the precursors of omega-3 we consume, will lead to the synthesis of the same amount of the long-chain fatty acids, especially the arachidonic acid that is accumulated in some phospholipids of several cells in the body, and for the omega-6 family, and also the EPA and DHA, docosahexaenoic fatty acid, that are accumulated in the body also. So the ratio between the long-chain omega-6 and omega-3 will depend on what we consume as precursors. But we can also eat directly this long-chain and find this long-chain in the food, and it's very easy to find arachidonic acid. It's almost everywhere, while EPA and DHA, the long-chain omega-3, are found in specific fish, for example, the oily fishes, but also in some algae. That is a new source for this omega-3. And what is quite important concerning this arachidonic acid, but also DHA, is that it's highly found in the brain, but also in the retina. And this is why we are highly interested in these two forms of PUFA. And so just to give you some numbers, 30% of dry weight of the brain is lipid, 30% are polyunsaturated fatty acid, and half is DHA. And DHA is the only form that is found in the brain, accumulated in the brain, while EPA is poorly found in the brain. And this is quite specific of the brain, as EPA is found almost everywhere in the body. And what is important to know concerning the brain is that this incorporation of both DHA and arachidonic acid occurs very early in life during development. As you can see here, it starts to increase in the brain during the prenatal period and also continue after birth. And so this occurs in parallel of brain growth, and this is found in humans but also in animal models. So we're working in mice mainly, and in mice we find exactly the same increase during brain development. And at a certain level, the DHA and arachidonic acid cannot increase anymore in the brain, but it has to be maintained. And of course, the quantity, the quality of omega-3 and omega-6 that are consumed are important to maintain this level during the rest of the life at adulthood, and also especially during aging, because the enzymes that are necessary to transform the precursors just have an impairment with age. So it's important to consume more long form of DHA during aging. So just in this slide, I try to summarize what we knew nowadays concerning how DHA reached the brain. So as I told you, the precursors of omega-3, the alpha-linolenic acid, is found, of course, in the food. Only 1.4% is converted into DHA, and this occurs mainly in the liver, and this is very important because the source of DHA coming from precursors is quite poor, meaning that it's very important to also have some DHA coming from fishes, oily fishes, or algae. Once this DHA is converted or consumed directly as a preformed DHA, it reaches the brain, so it is transported through the blood to go directly into the brain. And actually there is much debate on the way this DHA can reach the brain. Some claim that free DHA is able to just cross the blood-brain barrier and then go directly into neurons and glial cells, and others claim that there are specific receptors, transporters on the endothelial cells of the vessel in the brain. And actually one study recently published identified a receptor that is called MFSD2A, and that is specifically transport DHA when it is esterified to phosphatidylcholine, meaning that it could be that some forms are specifically translocated to the brain, while other, and especially the free pool, can just reach the brain freely. But it is not known whether this receptor is expressed during brain development. It's very important for this when the blood-brain barrier is not formed, especially during the prenatal period. Once this DHA reaches the brain and is incorporated into phospholipids of neurons, but also of glial cells, then it could be also metabolized into several derivatives. And the phospholipase A2 is a very important enzyme necessary to de-esterify this DHA out of the phospholipids. And then other enzymes are involved in the synthesis of many derivatives, and I will focus today on these derivatives, the resolvins, myrisine, and repotin D1, that have been shown recently to play an important role in the regulation of inflammation. And just of note, there is a specific enzyme, the ILOV4, that is expressed in the brain, and that is necessary to transform this DHA into very long chain PUFA with a very long chain of carbon, but it's fully known what is the role of this very long chain PUFA. So what is also very well known now, looking at the literature, even if some data show that there is no direct link between omega-3 and cognition, is that the metabolism of this polyunsaturated fatty acid is very important in mood and cognitive disorders. So there are many data showing an inverse relationship between fish consumption, so rich in DHA, and the prevalence of depression and cognitive decline. And also that there is a decreased level of omega-3 in the blood, but also in the brain of depressed subjects, so committing suicide in particular, but also of aged patients diagnosed with Alzheimer's disease or cognitive impairment. And of course, a lot of animal studies using dietary approaches show that the decrease of omega-3 in the brain alter emotional behavior, but also memory. But the question is, how does it work? Because it's important to notice that most of the studies in humans trying to do some supplementation with omega-3 failed and report poorly improvement in mood disorders, but also in cognitive decline. So I try to summarize here what is currently known concerning the cellular activity of PUFA. So as I told you before, the omega-3 and omega-6, so arachidonic acid and DHA, are esterified into phospholipids of membrane. So it confers to the membrane really strong potency in terms of activity. It influences, for example, RAFT, and RAFT are made of cholesterol and specific lipids. phospholipids, and the quality of omega-3, omega-6 that are found in the phospholipids change the position of receptors in the raft and also the way they are linked to the signaling pathways. It also influences the channel activity, and as I told you before, some of these long-chain fatty acid, arachidonic acid for the omega-6 family and DHA for the omega-3, can be deesterified of the membrane and then be metabolized into several derivatives. The prostaglandins, epoxide, and docosanoids that are very important in lipid signaling, but also in the regulation of inflammation. Some data show that these compounds can act directly on specific receptors. Some are nuclear receptors like ARXR, and also our receptors that are buried at the level of the membrane, and then influence directly gene regulation. And we have shown that it is also important in the regulation of endocannabinoids and synaptic plasticity through the activity of specific receptors, but I won't talk about that today. Concerning the inflammation, what is currently thought, but this is much more complicated, of course, because things are not so simple, is that the omega-3 are anti-inflammatory and pro-resolutive because they are precursors, and in particular DHA, of resolvins and neuroprotectin that have been shown by the group of Serran, but also of Bazin, to play a really important role in the resolution of inflammation, while the arachidonic acid is more considered as a pro-inflammatory factor. But I have to say that it's not so simple, because some derivatives have been shown through this pathway, the arachidonic acid, to be anti-inflammatory, and not all eicosanoids are pro-inflammatory. And also, as I told you before, arachidonic acid is a precursor of endocannabinoids, and these endocannabinoids regulate synaptic plasticity, but also play a key role in the regulation of inflammation. And some of these endocannabinoids are very important to downregulate inflammation. But keep in mind that anyway, the proportion of omega-3 and omega-6 is very important in the regulation of inflammation. And what is known, and we have been working for a very long time on trying to understanding the role of neuroinflammation in the brain, and what we have shown, and we and others, I mean there are many groups and some are here that have been working on the role of inflammatory factors, in particular pro-inflammatory cytokines that are produced in the brain in mood and cognitive disorders. And what is known now is that microglia, that is the resident macrophage into the brain, and you'll see that I will talk a lot about these cells, but because I love the cells. The cytokines, the pro-inflammatory cytokines in particular, interleukin-1, interleukin-6, and tumor necrosis factor alpha, can influence directly the synaptic plasticity and then be involved in mood and cognitive disorders, but also through their effect on some neurotransmitter, in particular serotonin and dopamine, and that are involved in mood. And the question for us is to understand whether polyunsaturated fatty acid can regulate this synthesis or the activity of microglia and then play a role in mood and cognitive disorders. I have to say that microglia are very important cells because they not only produce pro-inflammatory cytokines, but are also able to phagocyte specifically some spines at the level of the neurons and then play a role in the shaping of the neuronal network and the brain activity, especially involved in cognition. So these cells, as I told you, and here they are in yellow, are at the vicinity of neurons, and here it's an example of the hippocampus, and they really have a strong interaction with neurons, and some factors are involved to, so cytokines are produced by microglial cells and then influence neuronal function, but some also chemokines are produced by neurons and then inform microglia on what they have to do. And in the healthy brain, and especially during brain development, it is now known that these cells are very important in shaping the neurons, and the neurons just inform the microglia cells of what they have to do is they have to phagocyte some specific spines or not, and then really play a strong interplay, and there is a strong interplay between those two cells. So as I told you, the main question for us is to understand whether polyunsaturated fatty acid can influence so memory and emotional behavior. So I will show you some results obtained in animals. And so we just started by changing the level of omega-3 in the diet. But when we play with omega-3, we also play with omega-6, because what we want to do is to keep the total amount of lipid the same in the diet. So we have two kinds of diet. The first one is balanced into omega-3 and omega-6, and we use a ratio. We use only precursors, and the ratio is 1:5, and this is, at least in France, the recommended ratio for omega-3 to omega-6. And while the other diet we use is devoid of omega-6, of omega-3, meaning that it's highly enriched in omega-6. So of course it has nothing to do with real life. It's not, the purpose here is not to model what we eat, but really to change only the ratio of omega-6 and omega-6 and omega-3 and omega-6, and to analyze what is going on in the brain. So our bet was that if we decrease omega-3 level, we will impair brain lipid metabolism, but also the microglia, the brain innate immunity function, but also neuroplasticity and behavior, while increasing this omega-3 in the diet will promote this. And so I would like just to show, I have no idea, okay. So I would like to show you some results. First of all, so we use this diet, and this diet, as I told you, are really changed only in the level of omega-3 and omega-6, meaning that everything is the same concerning proteins and sugar and also vitamins that are very important to protect these fatty acids. And so the diet was given from the first day of gestation until so here in this case until weaning, and we measure at the level of the hippocampus the amount of lipids we could find in the hippocampus. And here, as you can see, so when animals eat this diet devoid of omega-3, there is a strong decrease in DHA, but no changes in arachidonic acid. So the DHA is compensated by the increase in DPA omega-6, and this is classical. This is obtained in most of the studies, and this is also found in humans. There is, when DHA is decreased, DPA-N6 is increased. And this is specific to the hippocampus, but when we look at microglia, so in this case we took out of the brain microglia, and we measure also the same fatty acid in these cells, we could find that in this case there is a poor decrease in DHA but a strong increase in arachidonic acid, meaning that it's very important to consider that not all the cells in the brain are impacted in the same way by the diets, and that it's quite important in order to understand the mechanisms underlying the effect of diets on in particular on neuroinflammation, to consider the amount in each type of cells. But trust me, it's not easy to do that. But anyway, what we did is that we just analyzed then by using 2 photons the activity of these cells. So microglial cells got processes, and they are moving their processes in order to connect neurons. I told you that they have a lot of connection with neurons, as you can see here. And what we found is that the deficiency in omega-3 lead to a decrease in the process velocity, and in particular the retraction, meaning that these cells were less able to move and to connect different neurons. They were sticking to one kind of neurons. And actually we also measured the number of microglia in the brain of these two animals, the hippocampus of animals submitted to the diet, and could not find any differences in the number of microglia. So it's only the velocity that is changed. Anyway, they also have a particular they express specific molecules, and here we could find that the deficiency lead to the increase of proinflammatory cytokine. Of course, this increase is very small. It's a tiny, tiny increase of TNF-alpha in particular. We couldn't see any differences in interleukin-6 and interleukin-1 beta, but other markers such as MCP1 and CX3CL1 that are chemokines involved in the relationship between neurons and microglia were also increased, suggesting that overall the interaction between neurons and microglia were changed. So what we did then is that we treated animals with LPS. So LPS is the toxin of gram-negative bacteria, and it's classically used to activate cytokine production by the immune system. And we found that the increase in cytokine production in the brain was higher in the hippocampus of animals that have been deficient in omega-3, suggesting that microglia are more prone to synthesize pro-inflammatory factors. And we also measured the behavior of these animals in just looking at social behavior, anxiety-like behavior. We also measured depressive-like behavior. It's quite difficult to measure depression in rodents, but we have some specific tests, and also spatial memory. And we found that overall they have an alteration in emotional behavior with a decreased social behavior, with an increased anxiety-like behavior. Here we measured the time they spend in the open arms in a specific test, and you see that this time is decreased, meaning that they are more anxious, and also spatial recognition. So we use a test that is a spatial recognition test, and when the demand is not too high, the animals seems to be normal, so meaning that the spatial memory is not impaired. But when the demand is higher, meaning that we ask them to really remember hardly what they have seen before, then they are impaired, meaning that the spatial memory is impaired in these animals. And we also looked at the size and the shape of neurons. Here you can see in the hippocampus that overall the neurons didn't we found differences in the length of the branches of neurons in the hippocampus, and this was accompanied by a change also in the number of spines. And as you can see here, the number of spines is at the level of the hippocampus is decreased in the brain of deficient animal, and this occurs quite early in life, as soon as weaning, and it persists later in life at adulthood. And also we measure the synaptic activity. So this is LTP here. We just activate neurons in the hippocampus. So you heard a lot about the hippocampus, and we measured the synaptic activity after this high stimulation. And this is a normal response. So this is called long-term potentiation. And you can see that in the hippocampus of mice that have been devoid of omega-3, there is no LTP at all, meaning that not only the shape of the neurons in the hippocampus is impaired, the number of spines is impaired, but the connection between these neurons is also impaired. We also found the same kind of results in the prefrontal cortex. So here you can see the shape of neurons in the controlled diet, and this is the shape of neurons of mice in the omega-3 deficient diet. So it's really very impaired. And again, the LTD, in this case, this is another kind of synaptic plasticity. So this is a normal plasticity, and it's totally impaired in the brain of the mice submitted to omega-3 deficient diet. But the good news is that if we give back some, at weaning, if we give back some fish oil, so rich in EPA and DHA, we can restore memory. We are just trying to understand now, how does it come? Is it because the neurons are good again? Are they plastic again, or is it because microglia are less pro-inflammatory? We don't know, and we're trying to understand this. So my time is running. I'm sorry. So just to give you another kind of result we had is that we tried to increase the level of omega-3 not through dietary means, but by using some transgenic mice, so the fat one mice. It was a gift from the Dr. Kang at Harvard, and these mice express a gene that is only expressed in C. elegans, and that is able to convert omega-6 into omega-3, meaning that when you submit these mice to a diet that is devoid of omega-3, then they are able to increase the level of omega-3 because they just convert omega-6 into omega-3. So this is quite convenient because we have only one diet, and we submit the diet to wild-type mice or to fat one mice. And what we found here is that spatial memory is, I'm sorry for that, spatial memory is impaired when animals are treated associated with LPS, and they have an impairment here in memory, and this is not the case, they have an impairment in memory, and this is not the case in the FAT1 mice. And just to show you what we found on microglia, actually the FAT1 mice got a phenotype of microglia that is pro-resolutive, meaning that they are less inflammatory. And this is also the case because they express less pro-inflammatory cytokines, more anti-inflammatory cytokines, and more BDNF. And you heard about this BDNF, that is a neurotrophic factor important for memory. And just to show you also the shape of microglia, so this is a microglia of old mice, and you see that it's very shrinky, and this is not the case in the brain of old FAT1 mice. So it suggests that the increase in omega-3 in the brain of these age animals are just very important in the modulation of these pro-inflammatory cytokines and the activity of microglia. So just to sum up, what I tried to show you is that when the diet is rich in omega-6 and poor in omega-3, then the microglia function is changed, is more pro-inflammatory, is sensitized to inflammation-induced memory deficit with a sustained neuroinflammatory factor, and we've also found some receptor dysregulation and impaired synaptic plasticity, while when the ratio between omega-6 and omega-3 is better, let's say, I don't know how to call it, but when omega-3 level is increased, then it protects from inflammation-associated memory deficit. I show you some example for LPS treatment, but we found the same in aging, and this is because it promotes a faster resolution of neuroinflammation with anti-inflammatory cytokines. And I would like to finish by just talking about translational perspective. I think it's quite important to define, before going for omega-3 supplementation, what is the inflammatory status, but also the metabolism of this PUFA in patients, because as I told you before, it's poorly convincing that supplementation with fish oil can improve memory deficit in Alzheimer's disease or even mood disorders, except that there are new data, especially in patients suffering of mood disorders. Only the one with inflammation got an improvement in mood disorders when they are supplied with EPA and DHA. So I think it's very important to understand first the mechanisms, and this is what I try to show you by using animal models, and then to translate to humans in taking into account the metabolism of this PUFA and also the inflammatory status. And I would like to thank people who did the work, especially young students and very nice people, so Quentin Leroy, Charlotte Mador, Jean-Christophe Delpech, Or Tomaso, Agnès Nadja, and Corinne Joffre. And I would like to thank you for your attention, and sorry for being so long.